All right, we're gonna walk through photosynthesis, and the way I'm gonna do this is first I'm going to draw a thylakoid, okay? Or I guess this is my grana. This whole stack is a grana. One of these is a thylakoid. We're looking at we're inside of a cell, okay? Inside a plant cell. So if we've got my cell membrane, my cell wall on the outside. Okay, so this is really, we're looking inside a plant. Okay, so I'm going to get rid of those. I'm actually making them a little bit closer. All right, so we know what we're looking at. When we are undergoing photosynthesis, we're going to look in two different locations. Okay, we're going to have the light-dependent reactions occurring in the thylakoids, specifically in the membrane of the thylakoid. So I'm going to show you that in a little bit, but we're also going to have the Calvin cycle, which is occurring here in the stroma. All right, so we have the Calvin cycle occurring in the stroma and the outside here. And all of this, the purpose here is to take sunlight, right, here's our sunlight. We're going to use energy from the sun in the LDR to generate glucose in the Calvin cycle, which is a form of energy that can be broken down by us heterotrophs, which do not make our own energy. Plants are autotrophs, as well as some bacteria, but we are heterotrophs. We use energy from other organisms. So we are going to zoom in on the light-dependent reactions first, okay? And for our light-dependent reactions, I'm just going to draw another, just a little double membrane here. So it's a little bit easier to see. Okay, so we've got our double membrane on our thylakoid. So here, in terms of, just to get our bearings of where we're at, here's our stroma. And here is the inner thylakoid. And we're going to go ahead and draw our photosystems, our transmembrane proteins. So first we've got photosystem 1. We're going to have an electron carrier protein. Then we are going to have our big cytochrome complex, another carrier protein. This is going to be photosystem 1. And then I'm going to have ATP synthase on the other side, okay, of the membrane. Still in the same membrane, but just kind of on the bottom. It kind of frees things up. But what is going to happen here is first we have sunlight, energy from the sun, that are going to hit photosystem 2. A photon is going to hit photosystem 2. This excites an electron in photosystem 2. It's going to pass this electron on to our protein carrier, and this excited electron needs to be replaced. So we are going to, what also is happening during this time is we are going to be generating oxygen and hydrogen. I'm going to draw those in a different color. Oxygen and H+ ions. In order to do that, we undergo something called hydrolysis, where we take H2O. All right, we are breaking water, hydrolysis, and we're using that energy to replace that energy, or to replace that electron. Now it's a low energy electron, but it's still an electron that then can be excited when we hit it with another photon, okay? So this process here is really important. We're breaking water. Ultimately, and what we're gonna say is water is being oxidized here. Okay, it's being oxidized, meaning it's losing our electrons, and we are going to pass those electrons down our electron transport chain. So we're gonna start sending this electron across the cytochrome complex. As it goes across the cytochrome complex, it's going to oxidize. The electron is going to be oxidized, meaning it's going to lose some energy. We're going to reduce our cytochrome complex. Energy is going into our cytochrome complex. Sorry, I drew that the wrong way. Energy is going into our cytochrome complex. We're going to pump more hydrogen ions to the inside, kind of like in cellular respiration, right? Where every time an electron passes through, that energy is used to pump protons or hydrogen ions across the membrane. Every time that happens, We're also going to pass more electrons across, and by the time they get to about cytochrome, or by the time they get to photosystem 1, they have pretty low energy. So they need to be excited again, so they're going to be hit by another photon of light here. Now that they have this high energy electron, we are going to, this process is called NADH reductase, or NADP reductase. So we are going to have our NADP, I'm going to draw that in blue, NADP. Now note that this is different than NAD, it's NADP, it's a different electron carrier, but we're going to use this electron, it's going to reduce NADP, and we're going to end up with NADPH. All right, this NADPH carrying this hydrogen and the energy in the form of an electron, they're going to be carried onto the Calvin cycle. All right, so we're going to come back to this in a little bit, but for now, we're going to shoot this NADPH across to the Calvin cycle somewhere else in the stroma. But we haven't finished the job yet, okay? So we're still pumping these hydrogen ions across our cytochrome complex, and we're ending up with a high concentration of H+ ions, right? So with our hydrogen ions, we want to use that to our advantage. And, oh, you know what? I drew my ATP synthase backwards. I'm going to erase that quick for you guys. So our ATP synthase is going to be our ticket out for those hydrogen ions. So I'm going to draw ATP synthase. I'm going to draw kind of a rotor on this side. All right, and this rotor, imagine that this rotor has ADP plus P on it. Okay, it's just kind of sitting in there. It's ATP synthase has ATP plus P kind of bound to it, so that when these hydrogen ions, as they're gaining more and more energy, as they're being pumped across or against their concentration gradient, they're going to passively pass through ATP synthase. Okay, and every time they go through, kind of like in cellular respiration, we're going to combine ADP plus P, generating ATP, our high energy molecule. All right, so we're generating ATP here, and this energy is going to go to the Calvin cycle as well. So I'm going to draw an arrow shooting it off to the Calvin cycle. All right, so here we have part 1, the light-dependent reactions. Note I'm going to get my highlighter out. I'm going to do this in orange. We'll do orange. With my light-dependent reactions, let's look at our reactants. All right, and we have light here. We have water. We have NADP. And we have ADP. Okay, these are our reactants. Now, what are our products? As a result of the light-dependent reactions, we end up with the following products: oxygen, ATP, and NADPH. All right, our oxygen is going to be diffused out of the cell. Our NADPH and our ATP are both going to go to the Calvin cycle. So let's go ahead and take a look at the Calvin cycle. All right, so we're going to be over here in our Calvin cycle. And in the Calvin cycle, I'm going to show you our reactant, which I'm going to zoom out so we can have, actually our cell's almost too big. So ultimately what's happening here is we have these really cool cells, or these openings on our cell membrane, our plant cell, and what's coming into these? We have carbon dioxide entering in, all right, into the cell. They open up, and it allows carbon dioxide to come in. It also allows oxygen to leave. Okay, that's just something that plants have. They have these stomata, if you watch the video with Joy and Shauna. But we are going to take advantage of this stomata that allows carbon dioxide in, and that's going to be our first reactant in the Calvin cycle. So now we can zoom in, sorry about that. Now we're going to start the Calvin cycle. I'm going to write that up at the top for you guys. Calvin cycle. All right, so for our Calvin cycle, our first reactant is going to be carbon dioxide, CO2. Okay, we're going to take a couple of these CO2. They're going to undergo part 1, which is called fixation. All right, fixation. Throughout this process, we are going to take RUBP and carbon dioxide, and we are going to combine them two. However, in this reaction that is happening, we're also going to see our friend Rubisco, which is really like a scissors that's going to cut. That's a really bad scissors, okay? It's going to cut our carbon dioxide and our UBP, and we end up with a molecule called PGA, which, again, don't need to know the specific molecules. You really don't need to know RUBP or Rubisco, but ultimately you're understanding that carbon dioxide, our main reactant, is being fixated into a molecule, into an organic molecule. Okay, it's being fixated from the air into an organic molecule. Next, we're going to undergo our second process. This is our first process, fixation. Our second process is called reduction. Reduction. Okay, so in reduction, what's happening is we're going to take our ATP from the Calvin cycle. I'm going to erase this arrow here if I can. Okay, I'm going to draw an arrow of it coming all the way over here, okay? And what's going to happen is this ATP is going to be oxidized. ADP plus P. Ultimately, we're donating that phosphate to our molecule here, but we're taking energy from ATP and using it to generate this high energy molecule from PGA. So I'm just going to represent PGA with some carbons here, and we're going to have our carbons here. I'm going to throw a P on there for you just to show that it's taking something from ATP. And we also are going to have NADPH coming over here. So I know it has an arrow pointing over this way. I want to keep it over there. So I'm going to have NADP. I'll just draw our arrow coming down here. Our NADPH is being oxidized, and we're reducing oil rig. Remember, reduction is gain. We're gaining energy, that's specific hydrogen, from NADPH, generating NADP plus, and we are going to, I'll just slap that hydrogen on the front of there, to create this high energy molecule called PGAL. All right, so now we have PGAL. It has a lot of names. You might also hear 3GP, G3P. And we are going to use this 3-carbon sugar, to produce glucose and other biological molecules. So I'm going to just pass this on, and ultimately what we're going to find from that is I'm going to draw our glucose. All right. Okay, this glucose, and what's cool about this glucose is that at this point it's got a lot of energy, okay? So ultimately we're producing biological molecules, but we're not done. High energy biological molecules. We're not done though. We need to finish our Calvin cycle, and PGAL, only 1 for every 6 PGAL are going to go on to become glucose. The other 5 are going to be regenerated to RUBP again, okay? And to do this process, we're going to take some of our ATP that was produced in the light-dependent reactions, and we're going to convert these ATPs to ADP plus P. In order to regenerate RUBP, we need to use energy to undergo this process. This is a series of reactions that's happening. So this is a really, really, really simplified version, but ultimately what you need to know is that ATP is being converted to ADP plus P here. Okay, so we have our Calvin cycle. Let's identify our products and our reactants, okay? For our reactants, I think I did orange and the other in the light-dependent reaction. So our reactants here for the Calvin cycle, we have ATP, we have ATP, we have NADPH, and we have CO2. All right. What are our products? Our products are ADP, NADP, and glucose. All right. Now, why is RUBP not indicated as a product or a reactant? It is going to be a substrate. I'm just going to say it's a substrate. All right. How you would need to know is that it is a substrate. It's not a primary reactant or product that you need to know. Okay. So looking at these products and reactants, what you can do now, I'm going to zoom out on the whole shebang. And I actually did forget to label this. My bad. Let me label this as the light dependent reactions. Light dependent. All right, so for light dependent reactions, we have our Calvin cycle. And what you need to know is how these two are working together. Okay, so ultimately we're donating a lot of high energy proton, high energy electron carriers to the Calvin cycle through our electron transport chain. And we're using these then to help generate glucose from carbon dioxide, all right? Really know the relationships, the inputs, the outputs. I'm gonna pull up our, I'm gonna do what I did for the other video. We're going to write our formula. So for photosynthesis, we have 6 CO2 plus 6 H2O plus light. Sorry. And we end up with ultimately C6 H12 O6 plus O2. Now, we also will talk about, sometimes you will hear water is being generated as part of this process. You don't really need to know that for AP Bio, but ultimately know that where is the carbon dioxide coming in, where's the water coming in, where's the glucose ending up, and where's the oxygen going, okay? And how are these all related to each other? What you can do is if you move on to the photosynthesis, the case study, which is really an actual experiment that was done on chloroplasts where they inhibit different parts of the process of photosynthesis of these two processes, and as they inhibit different parts of this process, they're able to investigate how herbicides can help target, or how we can use herbicides to target different parts of photosynthesis to prevent plants from growing. So I would encourage you to do that. This is an example that could very easily come up on a test or on an FRQ type situation, so I would encourage you to do it. It really concretes your understanding of photosynthesis. If you have questions, please ask. Otherwise, practice, practice, practice. This is one of those concepts that you just kind of need to drill into your brain, okay? Thank you for listening.